Hi everyone, my name is Adan Deeb and my talk today is on contest versus scramble competition, a comparison of feeding vibrations of seed parasite larvae. In natural ecosystems, resources are never infinite. Therefore, competition strategies have evolved in order to increase fitness, and these differ across populations. Seed parasites are organisms that use dried legume seeds as their host, and they cannot leave to forage for new resources if their host is depleted, so their competition strategies are uniquely important for their fitness. The first competition strategy that I looked at was scramble competition. This can be thought of kind of like an Easter egg hunt, where organisms will run around and gather as many eggs or resources as possible before they are depleted. This is an extreme on a continuum to contest competition. This involves direct conflict between individuals and usually ends in a winner-takes-all scenario. As you can see, these populations will compete differently for the same resources. Organisms are also under more competitive stress when population density increases, so higher population density will result in less resources and therefore more competition for these foraging individuals. The question that I aim to look at is, can feeding vibrations actually be used to approximate population density? And these feeding vibrations are vibrations that are generated by foraging parasites. I used Calisabruchus maculatus as the organism for my study, and in these organisms, the male and the female will mate, and the female will lay eggs on the seed, and then the larvae will burrow into the seed from the egg, where it will gather resources, and then it will pupate in the seed and emerge as an adult. And what's interesting about these organisms is that once they emerge from the seed as adult, they do not need any additional resources, so the resources that they gather as larvae in the seed directly affects their fitness as adults. So I first looked at some variables that could affect feeding vibrations. I looked at the age of the larvae when they were first detected, the time of day at their highest detection, and how long it took these larvae to respond after they had been disturbed. For this study, I used 20 beetles per competition strategy. I measured them from hatching to pupation to adult emergence, and I used 3 groups of beetles and measured each group once a day. So how did we turn feeding vibrations into sound waves? So this machine that you see on your right is our Munch-O-Matic that we built for this study. And the little yellow dot on the top, circled in red, is our piezoelectric guitar pickup. So you place the bean on the guitar pickup, it will pick up the vibrations from the bean. That will then go through this digital converter, which will convert the analog sound into digital sound. And that will then record the waveform on the computer, and you can plug your headphones into the digital converter and actually hear them chewing in real time. This is a waveform and soundbite that we recorded using our machine, and I will actually play the soundbite for you so you can hear the larva chewing. And so our results for this study were actually obtained from listening to these recorded soundbites. If you recall, the first thing that my study looked at was the age of the larvae when they could first be detected. So we found that both scramble and contest competitors could be detected at around the same age, and that males and females did not differ in age when these vibrations were first detected. And this suggests that the creation and the detection of these vibrations are not different. We also looked at temporal feeding patterns and found that time of day did not affect detection of vibrations. This suggests continuous feeding, and this is likely because they are inside of an opaque system, and so photoperiod does not affect them. We also looked at how long it took the larvae to resume chewing after they had been disturbed. So the y-axis on the graph that you see down below is the time to first vibration in seconds, and the x-axis is the number of days post-hatching. So for the contest population, males were slower to begin chewing than the females. And for the scramble population, females were slower to begin chewing than the males. However, at 20 days old, this trend reversed for both of the competition strategies. So our next step was to look at how well our device actually compares to known methods. So we are analyzing our recordings to assess these basic characteristics of sound. And we are comparing our piezoelectric guitar pickup to a laser Doppler vibrometer that was used in a paper by Geddes and Yak. And this vibrometer will shoot a laser out at the seed will vibrate the laser, and that's how the machine will pick up these vibrations, versus our machine actually picks up vibrations through a substrate. What we found was that our device had too much background noise for us to be able to analyze our sound files. So in order to fix this, we placed 2 piezoelectric guitar pickups back to back, and this will hopefully minimize background noise and work to amplify our vibration recording. In this way, we will be able to analyze our waveform as well as our sound bites and help minimize human error. What are our next steps? If you recall, the question that this study was originally looking at was whether vibrations created as a result of foraging can be used to approximate population density. And we have found that both competition strategies can be detected at the same age, that time of day does not affect their feeding patterns, that larval vibrations can be detected through a bean substrate, and that the competition strategies will differ in their disturbance response. We believe that if vibrations can be detected through beans and not just within a single bean, then they are likely a very good cue for population density. And so this picture on your right is an example of our next steps. So the bean on the bottom, circled in red, is empty and does not have any eggs on it. And the bean on the top has a single egg on it, and we want to see if the vibrations from the top bean will actually travel through and reach the bottom bean. We also want to look at correlations between population density and the intensity of these vibrational signals. So the increase in population density, which would mean more chewing larvae, should also mean an increase in the intensity of vibrational signals. And we want to see if larvae actually use these signals to allocate resources. And we want to look at how larval development, and therefore resource competition, affects adult competition. So if you recall, adults do not need any sort of additional resources in order to survive. So what happens within the closed bean system as larvae has downstream effects on the adulthood outside of the system. And we think that vibrations could be the link between population density and behavior. I would like to thank Russell Moore from Faded Line Studios for his help with the construction of the Munch-O-Matic device that was used in this study, as well as the people of the Fox Lab and Dr. Joseph Deeb for their help with the conduction of this study. Thank you very much for your time and attention, and I will now take questions.